at the control of breathing during exercise. So to start off, why does exercise impact breathing? So the schematic here below shows an integrative system between the lungs, the heart, and the muscles. So if you think about it, if we focus on the right side of that schematic, when we start to exercise, this gear on the right starts to spin a little bit more as we need more oxygen delivered to the mitochondria, and we start producing more carbon dioxide or CO2. And as a consequence of that, the heart needs to increase cardiac output to pump blood throughout our body so we can transport oxygen to the muscles and CO2 back to the lungs. And as a result, we need to increase our breathing to get oxygen into the muscle and CO2 out of the muscle. That is how oxygen is taken up and how we exhale CO2. So the oxygen we require is in the atmospheric air, and we need these systems to work together to get the oxygen into the muscle to produce energy and ATP. And with that, the increase in with that, we increase the rate of oxygen uptake, or VO2, so that we can increase the rate of its utilization in the muscle at the same time as we increase our breathing to deal with the CO2. So it's an integrative system, and it's a system that and it's a system during exercise that produces an increase in our CO2 output in the VCO2 at the mouth to keep at pace with the rate of at which we produce CO2 metabolically within the muscle. So there's a clear link between the increasing muscle work and the requirements to increase breathing to sustain the increase in the rate of oxygen uptake and excretion of CO2 at the lungs. So why do we breathe during exercise? Breathing during exercise imposes two challenges, as I kind of briefly alluded to it earlier. First challenge is that the CO2 produced must be cleared to prevent the arterial PCO2 and hydrogen ion concentrations in our blood from rising and to maintain it at near resting levels. And secondly, we also need to replenish the oxygen that is extracted from the blood and used by the muscles. And therefore, we breathe to prevent arterial oxygen pressures from falling and making sure that the delivery of the red blood cells to our tissues are fully saturated with hemoglobin full of oxygen. And hence these requirements are met only if alveolar ventilation increases in proportion to our metabolic rate. So in this figure here, we have a relationship of ventilation and a production of VCO2 on the x-axis. We can see that it's a 1-to-1 ratio. It increases proportionally. The ventilation increases in proportion to the CO2 production. And then after a certain level towards the end of exercise, we actually need to increase our breathing to get rid of the CO2 that we produce at higher exercise intensities. And so to do that, it's really important to consider the alveolar ventilation equation that we see in front of us here. And when we think about predicting how ventilation is going to change when we begin to exercise. Here we have ventilation on the left side as a dependent variable and three important independent variables that are related to ventilation. We have the need for CO2 clearance, or VCO2, the need to maintain arterial PCO2 around its set point that keeps our arterial pH constant. Our CO2 level and our acidity in the blood is tightly related to one another. And then, of course, we have the physiological dead space. And this is how much ventilation that we need to increase in order to maintain alveolar ventilation, which is specifically the part of breathing that happens, that participates in gas exchange at the alveoli levels in our lungs. So during exercise, the partial pressure of arterial CO2 doesn't change up to a certain point during exercise. So it remains around 40 millimeters of mercury and doesn't change, and we're very good at preserving it. And for the most part, the physiological dead space doesn't change a lot as well during exercise. And if anything, it actually slightly drops and we become more efficient in our ventilation, and the physiological dead space actually slightly reduces. So when we're predicting the breathing response, the most important factor that is responsible for increasing ventilation is the need to clear the CO2, or the VCO2 variable. Now, just to brush up on some definitions and that I'll be using throughout this lecture, and you've probably run into them in the readings. Throughout this lecture, there's two important definitions to consider. And the first one being hyperpnea, which is defined as an increase in ventilation that is proportional to metabolic demands, which is where the gases remain normal. So going back to this figure here on the right, we have ventilation versus VCO2. The hyperpneic phase, or the hyperpnea, exercise hyperpnea, is within these two colored regions, which I'll get into later. But you can see that the ventilation is perfectly coupled to VCO2, and the blood gases are PCO2, or arterial PCO2 remains around 40, and our pH, or our hydrogen ion concentration, doesn't really change. The second definition, we have the hyperventilation phase, which is an increase in ventilation that exceeds our metabolic demands, where blood gases deviate from normal, so overventilating and appropriate ventilation levels during exercise. But specifically for today, I'll be focusing on the hyperpneic phase of exercise and get into further details, examining or looking at the mechanisms involved during hyperpnea, and then we'll quickly touch up on the hyperventilation phase as well. And just going back to the figure here, we can see that in the hyperventilation phase, in this pink zone here, obviously ventilation drastically increases in proportion to VCO2. We can also see that the arterial PCO2 levels starts to fall, gets to around mid to low 30s, and we get more acidic. So our pH falls, which in relation, the hydrogen ion concentration increases. So how does breathing change as a function of exercise intensity? So before we talk about the control of breathing, we want to establish some further definitions and want to establish and understand how breathing changes as a function of exercise. And it's really important to, when talking about the control of ventilation during exercise, it's important to consider that in a given exercise intensity, there exists two metabolic boundaries, which results in three different exercise domains. So the first is the lactate threshold, which is the highest rate of work or the highest metabolic rate for that activity at which blood lactate can be maintained near resting levels. And this separates the the figure I showed earlier. This separates the moderate intensity, as we call it, from the heavy intensity exercise domain. And above that threshold, at a higher metabolic boundary, we have what's called the critical intensity, or the maximal metabolic steady state, which is defined as the highest exercise intensity at which a heightened lactate production in the muscle can be stabilized, so we can elevate our blood lactate, but eventually it will stabilize and it won't increase any further. And this separates what we call the heavy from the severe intensity exercise domain. So that blue region, or the purple region, from the red or the pink region. Now looking at this relationship here, so if we were to get on a bike in our lab or in any exercise physiology lab, doing an incremental exercise test, or if some of you have done a VO2 max test, with increasing the work rate, which increases the oxygen demand, the demand for oxygen will increase until we reach a point where you can no longer increase at which you can take up oxygen and utilize the oxygen in your muscle. And this would represent your VO2 max. And the schematic here on the bottom right is essentially a zoom in of the cellular level of the processes that go on in your cellular level. So like glycolysis, how we transport oxygen and CO2 in and out of the cell. And then we also have some blood flow here as well. So linking the heart to the lungs and the alveoli. And as mentioned in our previous slide, there are two metabolic boundaries. So if we were to start off from a very low VO2 at the beginning of the incremental tests with very low metabolic rate, and we gradually increase the work rate all the way up to our maximum, between rest and maximum, we will have the two metabolic boundaries that will reside. And all of us have it. So we must pass those metabolic boundaries and the different thresholds to get to our VO2 max. And we can detect these boundary crossing by measuring gas exchange and ventilation at the mouth, as well as blood gases or lactate. And that tells us a lot about why we breathe and why we breathe a certain way as exercise intensity increases. So putting some numbers into an incremental exercise test, we have some data from a representative participant here, where we can see that if we were to do exercise, we have some blood gas and the gas exchange variables presented. So we have ventilation, VO2, VCO2, the ratio of the ventilation to the metabolic production of oxygen, or of CO2 and uptake of oxygen, as well as end tidal oxygen and CO2 levels. And on the bottom here, we have some blood values measured, arterial blood levels. So what we can see is that at the start of exercise, our VO2 is pretty low. And then with increasing exercise intensity, we have an increase in ventilation and an increase in CO2. So you can see, if you take a closer look, you can see that, and you would appreciate that the ventilation is perfectly linear and coupled and goes at the same rate as the VCO2 as well as the VO2. And we can also see that between these ratios here. So if you look at the VE versus VE over VO2, you can see that it's a flat line. And then if you look at VE versus VCO2, you can see that that ratio is also a flat line. Another couple of points to consider here is that the lactate levels in the black dots at the bottom here is within normal levels. It's very low. It's just around 1 to 2 millimoles per liter. Our CO2 levels, our partial pressures of arterial CO2 is within resting levels, just around 40 millimeters of mercury. And you can see that their pH, or hydrogen ion concentration, is also very low. So this is what we call the moderate intensity, where we're starting exercise and it's a very light intensity if you've done a VO2 max, to put a perspective to it. Now, if we proceed with exercise and after we cross that first threshold, or the gas exchange threshold, or the lactate threshold, what we can see and what we can appreciate is that the VO2 is still increasing, obviously, but we have almost a break point in the ventilation. So the ventilation has an increase, or increases further with in respect to VO2. And we can see that with the VE over VO2 relationship, or the ratio here, we can see that after the first threshold, that ratio increases, but the ratio between the VE and the VCO2 is still coupled. So they're still increasing at the same rate, at the same intensity, and we can see that and appreciate that with this relationship here. The VE over VCO2, it's still a flat line. And this is still considered the hyperpneic phase. Obviously, we're into the heavy domain, or the heavy intensity of exercise, but it's still within, if you're exercising, it's still within a reasonable range where you can maintain exercise. You can probably talk to someone or keep up a conversation. It is a bit harder, and we can appreciate that with that increase in lactate. So your body is starting to produce lactate. Your blood is starting to get slightly acidic, so your pH is falling, and as a result, your hydrogen ion concentration is slightly increasing, and your PCO2 is still within just around 40 millimeters of mercury. There might be a slight drop, but it's pretty much negligible. Still around 40 millimeters of mercury, and this is still below the second threshold. So for the rest of this lecture, I'll be focusing on mostly, we'll touch up on the last intensity a little bit towards the end, but we'll be focusing around these two regions, which is defined as the exercise hyperpnea. Now, also just to add on, if you were to exercise at a constant intensity at the same relative intensity domains continuously, what we call the moderate domain in the green on the right here, we would see that over time, this individual was, I think, biking around 140 or 160 watts for 60 minutes. And we can see that there's no change in lactate. It's flat, and it's at a very low concentration, and it remains near resting levels. And there's also no change in the green here in the hydrogen ion concentration. It's around 37, 36 nanomolars. And similarly, the bicarbonate doesn't change as well. It's still a flat line. Now, focusing on the heavy intensity in the blue, we can see that there's an increase in lactate. The exercise intensity is a bit more challenging, but we're able to maintain that intensity, and the lactate doesn't really drift higher or increase any further. And with that, we're actually able to buffer that lactate through bicarbonate. We also have a decrease in bicarbonate. And then with that, we also have an increase in hydrogen ion concentration. So this is, like I said, the hyperpnea phase of exercise. So then the question becomes from the controlled breathing perspective is how do we know how hard we have to breathe and how is it proportionate to CO2 or the oxygen requirement of the muscle? So if we look at on the graph, we obviously see that it's very precise. But if we also look at this figure here that was published from our lab in 2022 by Ward and colleagues, if we were to have participants exercise from 20 watts and everybody starts, or all these conditions start at 20 watts, and then we bring them up to different intensities. Sorry. We go up to 60, 80, 100, and 120 watts. We can see that the VCO2 in the white dots is increasing with exercise intensity. And with that, ventilation is perfectly tracking and is perfectly coupled to VCO2. And this has really kind of got our attention and obviously gotten a lot of other researchers' attention, specifically also Dr. Duffin's. And we're really curious as to why that is. Why is it so tightly coupled? And but at those intensities and as well as our blood gases don't change. As we saw with the previous graph, our blood gases are around 40 millimeters of mercury for CO2, and lactate is within normal ranges or slightly higher, but it's really a puzzling question. And the goal from here on is to focus on some mechanisms and to see if we can explain some of the reasons that it might be. So for years, researchers have tried to understand what factors influence our breathing during exercise, and this diagram by Forster and colleagues in 2022 describes a nice schematic of the potential mechanisms involved during exercise, and we can summarize these into three potential groups or three sets of mechanisms. The first being the neural forward mechanisms or the central command, and this comes from our cortical regions. And the idea is that our brain uh, precisely or simply knows how much we need to breathe for a given activity that we're doing. It happens to naturally or automatically send neural signals that is all integrated and sorted out at the cortical level. There's also a lot of different feedback mechanisms. So two and three that have been reported to help drive ventilation. One of the mechanisms is the neural feedback mechanism. So this is within the muscle. There are receptors that are sensitive to muscle tension and stretch, and also to the metabolic state. So how acidic the muscle gets within the muscle itself that are known to send signals back up to the central nervous system that would drive ventilation and increase it and provide a negative feedback loop. And then, as you probably know and heard from previous lectures, there's also the arterial chemoreceptors, which sense arterial and central chemoreceptors or respiratory chemoreceptors, which sense falls in oxygen or rises in arterial hydrogen ion concentration that trigger a reflexive response to increase ventilation. And it is perceived and thought that all these mechanisms work together to participate in the control of breathing. And then from now on, I'll present a few papers looking at each specific mechanisms and try to understand what the researchers did to kind of prove each mechanism and how they were important to breathing during exercise. So first off, we have the feed-forward factors, our central command. This study, a landmark study published in the Journal of Science by Elbridge in 1981, was a pretty complicated experiment that was done in decorticated cats and neuromuscularly blocked cats. Or in other words, all the sensory afferent activity was removed from these cats. So they wanted to see what is driving or whether the central command mechanism is valid or not. And what they did was they wanted to get rid of afferent input from the blood and the afferent input from the muscles and see how that would impact breathing. So what they did, as you can see in the figure here, is they stimulated the central nervous system associated with motor activity in these cats in the brain region, and then they recorded from different muscles, and they recorded from the phrenic nerve. So they recorded from the left and right bicep femoris nerves, as well as the phrenic nerve activity, which is indicative of ventilation. And then they also had resting data and also fictive locomotion So this cat wasn't actually moving, but it thought that it was moving. So you can see here that we have, yeah, like I said, the left, the right bicep femoris, the phrenic nerve activity, as well as airway pressures. And you can see that when they made the cat, when they stimulated the different regions in the brain and they made the cat believe that it was actually exercising, it started to send signals to its muscles and have an increased activity of the nerves, innervating of the muscles of the cat. And you can see that there's also an increase in the peaks as well as the frequency of the phrenic nerve activity, meaning that there's more tidal volume and breathing frequency increases. And we can see that change from rest to the fictive locomotion state. So what this showed is that if we essentially began to breathe more during stimulation of these motor centers, they're independent of any humoral or neurogenic feedback, which supports the idea of central command, which is very important in determining the ventilatory response to exercise. All right. Next up, we have a looking at neural feedback. So this study was done by Aman in 2010 in the Journal of Applied Physiology, and this provides some evidence that afferents within the muscle nearly help support ventilation during exercise. So in this study, this experiment, they had individuals exercise at 4 different intensities. So a few minutes at 50 watts, 100, and 150 watts. And then they also had them exercise at near maximal levels. So near their VO2 max at 327 watts. And they did this over 2 conditions. The first condition was the placebo condition. So they brought these participants in and they injected, before they got on the bike, they injected into the interface of the lumbar spine with saline solution, and they had them exercise, and they quantified the gas exchange and the ventilatory response. So in the top panel, we have that VE, or the ventilatory equivalent to VCO2, VE VCO2 ratio, and then the end tidal CO2. So this is the response we get. It's pretty flat until they get to maximal exercise. It starts to increase. Similarly with end tidal CO2, it'll be slightly different than arterial CO2, but it's still relatively flat until they start to hyperventilate. And in the second condition, in the intervention condition, what they did was they injected fentanyl into the spine. And what that does is it blocks group 3 and 4 muscle afferents and blocks all the operating signals coming from the muscle to the brain. So the hypothesis was that if the afferents were important for breathing, that if we when proceeded to block these afferents, that should lower ventilation at a given metabolic rate. So these participants obviously did the same exercise protocol, and you can see in the second condition, the ventilation fell relative to VCO2. That's evident in both figures. We have a lower VE over VCO2 ratio. So there was an indication of hypoventilation, which is also evident in the end tidal CO2 response, because since we're breathing slightly less than the other condition, for the given demand of VCO2, or for a given demand of CO2, that's going to actually slightly increase the partial pressure of CO2. And you can see that that all exercise intensities ventilation was lower, even though VCO2 was exactly the same. So the same metabolic rate, but a lower ventilation. So therefore, the blocking the muscle afferents blunted the hyperpneic and the hyperventilatory response, suggesting that at least that they contribute partially to the ventilatory response to exercise. They obviously still breathe, but they don't breathe in proportion and to the need to eliminate CO2 as well as they did in the placebo condition. Next up, we have another pretty cool study. Probably one of my favorites is a study that was done looking at neural and humoral factors, done in 1963 by Cow. And this needs studies because they essentially, what they did was they swapped, they tested 2 dogs and they swapped their venous blood. So in the top panel, we have dog number 1, which is a neural dog. It was an exercising dog. And dog number 2 was a humoral only dog. And they were not, that dog was not exercising. So what they did was they swapped the blood So the neural dog venous blood was connected to the humoral dog, and the venous blood from the so that venous blood from the neural dog was actually entering into the humoral dog, but the humoral dog wasn't doing any exercise, and its venous blood was getting put into the neural dog. So in the first condition, or in dog number one, the neural dog, its limbs were electrically contracted. So and it was actually performing exercise, and you can see that it began to increase ventilation. So VE increased by about twofold, went from about 3 to 6 liters per minute. Um, and began to increase ventilation. But what was more important is that it was being perfused with the blood of the dog that wasn't doing anything, that wasn't exercising. So you can see that the VO2 didn't increase because there's no reduced oxygen coming back to the heart. But despite the lack of the humoral feedback or the evidence of the exercise of the neural dog, we still saw an increase in ventilation, which would be completely driven by the neural feedback or the neural activities from the muscle. Now, looking at the second dog, that was actually, or so looking at the dog that was actually exercising with the deoxygenated blood, so the neural dog's blood, which was deoxygenated, was transferred to the humoral dog, and it sensed that increase in VO2 even though it was not doing any exercise, and it had to increase ventilation. So we can see that as well here. Ventilation increase as well as the VO2 increase. So to summarize this means that in the absence of any central command or any humoral feedback, the neural dog had a ventilatory response, which would presumably was driven only by neural mechanisms, and the humoral dog, which wasn't doing any exercise, and didn't have any neural feedback as its muscles weren't moving, and it had no central command because it wasn't performing any exercise. Just by providing a humoral stimulus, it had an increase in ventilation. So what this suggests is that both neural and humoral factors are responsible or can be responsible in the absence of the other two factors to increase ventilation. Uh, Next up is a study that was part of my thesis, one of the first studies I did in our lab, and we wanted to examine the contribution of the peripheral chemoreceptors and its changes with increasing exercise intensity during exercise hyperpnea. So again, we're focusing on the hyperpneic stage of exercise. We brought participants in, and they cycled that during steady state of exercise at 4 increasing intensities. So we had them at rest. We quantified some measures at rest, mod 1, which was the first, mod 2. So these two intensities were below that lactate threshold. And then we also tested them at the heavy condition. So in between the first and the second threshold, where things did increase slightly, so they're still able to maintain their blood gases relatively. And then we wanted to quantify the contribution of the peripheral chemoreceptors. So the way we did that was at each intensity, we administered 100% of oxygen. So they were connected to a mask, and then we fed in 100% oxygen to diminish or to lower the carotid body contribution for about 30 seconds. And then we quantified the drop in ventilation from room air compared to the hyperoxic condition. And this would assess the tonic contribution of the peripheral chemoreceptors. And we can see that with increasing exercise intensity from rest, there's an increasing drop in ventilation during the hyperoxic gas administration, which was statistically significant. P is equal to less than 0.01. Another thing that we did, which was pretty cool and pretty novel for our lab, we also, during exercise, we sampled blood or arterial blood to measure blood gas levels during exercise. You can see a little schematic here. We had a participant, we would insert a catheter, and we can simultaneously, as they're biking, we can draw blood and actually analyze it for blood gases in that moment. And what we saw in the figure here from that data is we saw that, as expected, hydrogen ion concentration increased with exercise intensity. It was still, within normal range in the first 3 conditions here at the 20 watts mod and mod 2, mod 1 and mod 2. And it, as expected, it increased slightly in the 2 heavy conditions. And what we saw is that we were able to maintain, or the participants were able to maintain their arterial CO2 content, but it slightly dropped in the last condition to about 38 millimeters of mercury. And what we did was to see if the peripheral, to see what the driver or the stimulus for the peripheral chemoreceptors are during exercise, is we quantified, we statistically quantified and or used it. Let me backtrack here. We statistically used the hydrogen ion concentration increase as a covariate in our analysis. So if we were to use the hydrogen ion concentration in an ANCOVA model, so as a covariate, what was cool is that the significant effect that we saw using an ANOVA without the hydrogen ion concentration, which was significant, did not show any more significance. So the p-value went from 0.01 to 0.52, which would explain that the increased contribution from the chemoreceptors is reflective of an appropriate reflexive response to increases in circulating hydrogen ion concentration. So all those factors are important, but one of the things that obviously I mentioned earlier is that we still don't know why ventilation increases and is so tightly coupled to an increase and in proportion to VCO2. Obviously, we know that during incremental exercise, we showed that ventilation is perfectly coupled in proportion to VCO2. And again, this is the data that we collected in around 2022 by Ward and colleagues. We can see that ventilation perfectly was matching and following VCO2. But we also quantified the kinetics of the ventilatory response. So how fast our ventilation, our VCO2 increased for an exercise stimulus. And we can see that on the right. So we quantified the time constant, which is with these different intensities. And we knew that this would provide an indication and then potentially an explanation as to what is potentially going on. So when we compared the time constants of VCO2, so how long it took for the VCO2 and ventilation to reach steady state versus how long it took, for how long it took for the VCO2 and the ventilation to reach steady state. In the black and ventilation, and the open white circles we have VCO2. We can see that that on average they were well coupled, and you can see that the profiles are overlapping on one another. But what was really interesting was that ventilation was slightly slower and was slightly about 2 or 3 seconds lower than VCO2, which would potentially explain and indicate that the cause or the stimulus is VCO2 to ventilation. And the response to ventilation is something related to CO2 and must be important in determining the ventilatory response. So, but when you think about it, the CO2 really has no explanation from the neural feed forward in the literature, neural feedback or humoral feedback mechanisms that can explain this link between CO2 to ventilation, because we don't really have a way of sensing VCO2 necessarily in our body. So the study up next is an example of how well coupled ventilation is to VCO2. The study done in 1981 by Philipson in the Journal of Physiology is they wanted to look at and titrate VCO2 down in a sheep model and look how ventilation responded. So what they did is they maintained arterial hydrogen ion concentration constant. But what they did was arterial hydrogen ion concentration constant, but what they did was they manipulated the CO2 return on the right side of the heart. So if you have a higher CO2 return, that means there's a higher level of ventilation, a higher VCO2. And when they did this, these sheep, the sheep weren't exercising, they were at a rested state. And they had an increase in ventilation as they dropped the CO2 and the gradient. And the gradient is going to lower the VCO2. As they artificially lowered the VCO2, ventilation continued to drop in a linear manner to the point where they almost stopped breathing, and an apnea occurred in ventilation. It went to zero. So this would explain potentially the the coupling between the ventilation and the VCO2, that ventilation is related to VCO2. So looking at this slide here, just a quick model of some of the variables that is measured during exercise. On the left side, we have ventilation, tidal volume, and breathing frequency. These are the controlling variables of ventilation on the left, and the controlled variables on the right column here are the blood gases as a function of VCO2. So arterial CO2, O2, pH during the hypertonic phase of exercise. We can see that all of these really remain stable, but there's really no error signal within the blood that's related to to the CO2 that be driving this response. And one of the things that we think that might be plausible is that there's some sort of a mixed venous chemoreceptor on the right side of the heart. So this would provide an error signal proportionate to VCO2 that would drive our ventilation. And then we kind of saw some evidence there with sheep, and there's also some studies done in the 60s that have also showed potential evidence for mixed venous chemoreceptors. So this really remains a mystery of how tightly coupled or why ventilation and VCO2 can be so tightly coupled to one another. So obviously, so far we've focused on exercise below the critical intensity or the maximal set of metabolic steady state or the hypertonic phase of exercise. Now we're going to look into some data and some evidence above that intensity in the severe intensity or the hyperventilatory phase of exercise. So if we go back to this graph that we collected from this participant, we had them go all the way to their max. Once we transition from the second or past the second threshold, the respiratory compensation point, or above the maximal metabolic steady state, you can really see and appreciate that the ventilation skyrockets upwards. And at this stage of exercise, again, if you've done a VO2 max, you're essentially sprinting, you're panting, you're breathing really hard at really high intensities of exercise. And the reason we're doing that is we're trying to get rid of that as much of CO2 as we can in order to prevent from the hydrogen ion protons and the acidity in our blood from the lactate from rising to really high levels. So ventilation increased much more significantly in proportion to VCO2. You can see and appreciate that in that VE-VCO2 ratio here. You can see it's very flat up until a certain point, and then it just shoots up. Our our lactate levels goes through the roof. Same with the hydrogen ion concentration, and then we're no longer able to maintain our CO2 levels. So we go from about 40 to low to mid 30s of millimeters of mercury for partial pressure of arterial carbon dioxide. And this is called the respiratory, or that threshold, the one that we just passed, it's called the RCP, or the respiratory compensation point, and this is referred to as the hyperventilatory phase of exercise. And similarly, if we were to look at it at a constant power intensity, see if they can bike at a same intensity in that severe domain, in that red region, you can see a lot of differences. One being that they're not able to maintain, or they were not able to last the full 60 minutes of exercise. They only went for about 30 minutes of exercise, and you can appreciate how much higher the lactate is, how much higher the hydrogen ion concentration is, and we have also a massive drop at with our bicarbonate levels as well. So we're past the second threshold, and the hyperventilatory phase, we're not able to constrain the metabolic acidosis within this domain. So this is called the exercise hyperventilation. And just to reiterate, if you look at the relationship with VCO2 after the second break point, after we hit the RCP level, the hyperventilation stage begins, and we're no longer able to maintain blood gas levels. Yeah, so like I mentioned, one of the reasons that we believe is a mechanism involved in this significant ventilation increase during the hyperventilator phase is the carotid body, or the carotid chemoreflex. So again, hydrogen ion increases, or pH falls quite a bit, um, and our PCO2 falls, and it makes our carotid body sense and provide a reflexive response in our ventilation. It's going to increase the increase in the hydrogen ion concentration is going to increase ventilation to help constrain the metabolic acidosis. And a couple of other potential mechanisms is we also have an increase in norepinephrine, an increase in potassium concentration, adenosine, angiotensin, and temperature, which is which is going on at this stage of exercise. And one study that I want to focus on, which demonstrates the role of the carotid body in this process, is a study done by Mayer in 2004. What they did was they looked at an incremental exercise, and they again did it over two conditions. One being that they infused participants with bicarbonate or sodium bicarbonate near the respiratory compensation point. So just before the hyperventilatory phase, they would inject participants with sodium bicarbonate, and the other condition was, of course, placebo. So in the top panel here, we have pH over time, and we can see that the intervention worked, and they tested 5 participants, and they prevented a fall in pH following a sodium bicarbonate infusion, and this would abolish the hyperventilatory phase, or the response that we see during exercise. So the black dots is the saline solution, and then the open white circles is the bicarbonate injection, and you can see that they were able to prevent the hyperventilatory response and abolish it at the higher exercise intensities. And this provides good evidence that the respiratory compensation point and the hyperventilation is due to the reflexive response of the carotid body and the peripheral chemoreceptors. So just to wrap it up here, the simple answer is I don't know what's causing, and maybe a lot of people don't know, or we all have ideas of what's causing, or what are the mechanisms behind exercise hyperpnea, and specifically the precise linkage between ventilation and VCO2. And there's a couple of quotes here, one from experts in the area, from Ward and Whip, being that although many mechanisms have been demonstrated which can increase ventilation during exercise, the essential challenge which remains is why does ventilation only increase to levels commensurate with the level of pulmonary CO2 exchange. And another quote from Dr. Jerome Dempsey, one of my mentors, is he describes exercise hyperpnea as the ultimate mystery and something that still keeps him up at night and doesn't let him to go to sleep in peace. So that's it from me. Thank you very much, and hope